All right, everyone. So in this last lecture video for this series of four, looking at Mendelian genetics, the last thing that I want to talk about is meiosis. So the reason why I want to go into talking about meiosis is that a lot of the variation that we're seeing and what we end up looking at with Mendelian genetics are the products of what occurs during this process. So we already know what happens stepwise in the process of meiosis, right? We know some of the major events and we know what that can lead to in terms of the outcomes or probabilities of each of the gametes, right? We've done that with our Punnett squares now. We've looked at that just thinking about the process of segregation. Okay, now we're just going to kind of highlight a couple of those features again, just to reiterate where that variation comes from in organisms that are reproducing with a process like or using the process of meiosis. So again, let's think about what are the products of meiosis. So remember, for meiosis, what do we get? We get four daughter cells that are genetically different from each other and have one half of the genetic material. The number of divisions for meiosis, right? Remember, there are two, right? We have meiosis 1 and meiosis 2. And then with meiosis 1, that's the reduction step. That's where we're actually splitting the number of chromosomes in half. And then lastly, in meiosis 2, that's where we're taking the product from meiosis 1 and splitting the sister chromatids so that then we have the correct distribution of alleles, or correct distribution of chromosomes, sorry. And then what's the structure that forms around meiosis that doesn't occur during mitosis? So that, remember, is the tetrad, right? The point where we have the four different chromosomes that come together. So the homologous pair, right? The one you get from the mom, the one from the dad, coming together, crossing over, swapping genetic information, and then pulling themselves apart during anaphase 1. So remember, we talked about crossing over. And so again, this is the exchange of where you have the chromosomes, the homologous ones. So again, homologous chromosomes code for the same genetic information between those homologous chromosomes that result in a mixture of the parent's characteristics. So what that allows us to do is to take, a gene from from the parent, a gene from another parent that were together in the offspring, and then start swapping those around. Okay, and so when we say swapping, we're swapping the genetic information at the same, essentially, places on the chromosome, right? As we read it, it's sort of like a chapter in a book, right? You're just taking chapter 2 from one book and putting it in for chapter 2 in the other book, and then you're swapping out chapter 2 from that book and putting it into the other one. Okay, so, it might change the story a little bit, but for the most part, the same things are going to be there. Okay. So this process of crossing over, remember, takes place during prophase 1, right, as we're moving into metaphase 1. So as those sister chromatids come together to form the tetrad, right, we can swap those extension pieces, the arms of the chromosomes, over. So this is just a visualization of that process. So again, we have our homologous pair, we have the set from the mom and the set from the dad, and then once crossing over occurs, notice again that it's happening at the same places on the chromosome, and it's only going to happen at 1. It doesn't mean that crossing over is only going to happen here. It could also happen up here, and you can get crossing over occurring in both of those places. And then that leaves you with chromosomes that are different, right? So that each of the gametes has some genetic difference from each other. Right, this one and this one have a lot that are coming from the first parent, the blue parent here, but this individual has some of the genetic information that is coded from the purple parent. Okay, so they're going to get swapped and traded. So again, with crossing over, you're going to get this recombination of different alleles, right? And then once those genes are crossed over, the chromosomes and the chromatids may be exchanging genes. Okay, so after our first division in meiosis 1, we separate those homologous pairs, and then lastly, we separate the sister chromatids so that we get 4 haploid daughter cells. The other thing that's happening, again, why we get this genetic variation is due to the independent assortment. So after crossing over occurs, and you have individual chromosomes in all of these, just depending upon how they line up on the midline or that metaphase plate in the cell, it's going to dictate what the final combination of alleles available to the offspring are. Okay. So one of Mendel's principles, it states that basically within independent assortment is that the genes from different traits can segregate independently during this process, right? This is why he could get true breeding plants that were, say, green and tall, or that had green seeds and were tall, because those particular traits ended up segregating, right, because they were on different chromosomes. So whatever code was happening for that one versus the other could be partitioned and separated out. So it increased the probability of that likelihood of those particular traits coming together, right, because of the process that was happening. So again, with independent assortment, not only do you get from crossing over in tetrads. You have it also happening as they line up during any phase in meiosis, so when they're lined up for metaphase. So just even the order they line up, the direction they line up, depending upon which side the chromosome gets to. Like for example with this one, right, if this was here in the middle versus this one as they started to line up, it's going to change how and where the cells are going to be. Okay, so just because of how they line up, we can get a lot of variation. So this right here is showing you that probability, right? So here we have our individuals where we've got our homologous pairs lined up on the midline. And here we just happen to produce offspring where the homologous pairs are made up majority of one parent versus the other, right? That you're getting allelic combinations that match a lot of one of the parent. Here you're seeing more of a mixture, right? Just because these two chromosomes have lined up opposite of each other versus where they're in parallel with each other, we can get offspring that have now a combination of different alleles. So even here, just because of the probability from these lining up in a particular way, there's probability here, or the outcome here is essentially you could have 8 different possible ways that your offspring could get different allele combinations. Okay. The other thing that I wanted to talk about in this, and it's a little bit of an extension of what's happening in meiosis, is regarding a locus. One thing that we ended up finding out about Mendel's choices for traits is that a lot of the traits he didn't actually end up looking at were because they were on the same chromosome and he couldn't get them to segregate separately, except for one trait. There was one trait that's actually on the same chromosome, but it behaved like a segregated trait that was on different chromosomes because of the location on the chromosome. They were so far apart from each other, like all the way here versus all the way here, is that when the crossing over process occurred, it ended up occurring so often that he was seeing this particular characteristic show up over and over again. So it was just kind of weird luck. But what you would need to know about a locus is that a locus on a chromosome is a specific location where that gene is. Okay, so you can think of it as the address of the actual cell. Each of the gene is that the gene has a location it will always be found. So for example, this P gene, all right, might only be found on chromosome 3 of, say, our 46 chromosomes, and then it's going to be in a particular location near 2 other traits that we're able to identify, or 2 different genes that are able to identify, right? And we can use that information to help us. Okay, so the reason why knowing a gene locus is really important is that we can use that information to see how different traits get passed on, and it also can help inform a lot about genetic disorders, and that oftentimes if these locuses get switched or they get moved or they jump, so you might see that happen during an era of meiosis where crossing over flips things, they can get inverted and stuff like that, is that we can trace those locuses, and if they're in different places, that can often be a sign of another genetic disorder. So knowing the locus is important because we can actually track how different traits interact in an individual. Like if they're exhibiting this particular characteristic and they have this other characteristic, we can essentially use that information to determine what kind of mutation occurred. We could use it to track historical things, such as like with pedigrees, right? We can use a lot of that information and the nuances of it to allow us to compare many different things. So we can track 2 or more characteristics that interact, and we can use that information to help figure out how they get passed on. So nowadays, as we get more specific about crossing in, say, plants to produce food, oftentimes we can look at the gene maps and we can look and see where all the locuses are, and we can encourage cells or select for individuals that have these locuses that have been in particular places to get pure breeding lines that exhibit the beneficial traits that we're interested in. Okay, but we can also use that to determine genetic diseases and disorders based on what they have. So what you're seeing here is, again, the whole process of what's going on with the locus and why it matters where things are. Okay, so depending upon what physical characteristics the parents have, we can actually trace based on what physical characteristics the offspring have, what combinations occurred during crossing over, or what combinations might have arose in the cells due to independent assortment. So we can use all of that to help inform kind of the passing of traits. So I know plant breeders will look at this in order to produce flowers that have particular colors or shapes and things like that. People that study genetic disorders, gene therapists, for example, will trace that, and they will look at the probability of certain outcomes and the likelihood that you might get various combinations of alleles to help inform what is going to happen for parents, especially if they know that they carry a particular disorder. Like, what's the likelihood that this thing is going to show up in their offspring? So what you can see here is this is the gene map for a few of our genes, particularly chromosome 18, 19, 20, 21, and 22. So these are the shorter chromosomes in the human genome, and you can actually see on here where we have tracked and found a bunch of different genes. So all of these different genes have some kind of letter makeup that describes what the function of that particular gene is, like what does it produce, what protein does it do, and then a number that goes with it. And that number code is just to give us an idea of kind of where it's located. Excuse me. Oh, it's really late. To see where your chromosomes are located, and it's just kind of an identifier, again, like an address. Okay, so that's the end of our discussion of Mendelian genetics. So if you've got questions, let me know.